be that you're going to be the one presenting because you're going to pronounce it properly for more part.
I might mess up some of the French names though, so we'll see. I'll give it my best go. Okay, hello everyone. Welcome to today's CGIL seminar. Today we're delighted to have Audrey Martin, who is one of the PhD candidates here at Guelph, working under the supervision of Dr. Flavio Schenkel. Audrey received a degree in biological engineering from the University of Lyon in France in 2014 and a Bachelor's of Science specializing in livestock genetics from the University of Limoges, France in 2015. She did her undergraduate thesis on reproductive physiology under the supervision of Dr. Stephen Butler at the Chagas Moore Park Research Centre in Ireland. She then completed a Master's of Science in Genetic and Biodiversity at Wageningen University in the Netherlands in 2018, with the guidance of Dr. Marinus de Paas and Henry Wolders. Her thesis aimed at understanding the genetics of the estrous cycle in dairy cattle. During her master's, she came to CGIL as a visiting student under the supervision of Drs. Filippo Migliore and Louise Brito. She was working on estimating genetic parameters of methane emissions in dairy cattle, and since 2019, Audrey's thesis focuses on improving the genetic selection for reproduction in dairy cattle. And with that, I'll pass it over to you, Audrey. Thanks.
Thank you. Okay. This, and let me just the laser so you can see the pointer as well, right?
Yes, thank you.
Okay, thank you. Okay. So thank you for being here today. I'm Audrey Martin, as presented by Colin. I'm a PhD student for Dr. Flavio Schenkel at the University of Guelph, and my thesis is mostly on finding ways to improve selection of fertility in dairy cattle. Today, I would like to present you two of my projects, which are relatively different ways to reach this goal. So for each project, I will first give you some background on the topic, why is it interesting for the dairy industry, and then what was done generally. I will start with gametic incompatibility, which is a non-random fertilization event. First, I will explain you what it is, why we thought it could affect the reproduction ability within the dairy industry, and then I will tell you how we investigated it and show you which results we got from the analysis. So to explain you a bit about this term, I will go very general and then I will go more into the details to tell you why we are thinking it could be interesting for dairy cattle. So first of all, when we look at the definition of fertilization in our dictionary, it is about the unions of the gametes to form an embryo. This is very general, and if we take a more genetic perspective of it, we can think of the Mendelian laws of inheritance, and more specifically about the laws of segregation, which state that parental alleles are randomly segregating between gametes, and that those gametes have the same chance to be producing an offspring. Well, this is not always true. First, we know that parental alleles are not always randomly segregating, but the part that we are most interested here, in fact, is that not all gametes have the same chance to be transmitted. So here is the concept of non-random fertilization. You may think of it as a key and lock mechanism. If someone successfully reaches a door, it doesn't mean that they can enter. If the sperm cell has the ability to travel through the female reproductive tract to reach the egg, it doesn't mean it will fertilize the oocyte. There could be many reasons for that, and when those reasons are not associated with infertility or subfertility of one member or both members of a pair, we could be looking at gametic incompatibility. This is a very recent topic in human research, where it is challenging the definition of infertility, as it is not a disorder of an individual within the couple, but basically an incompatibility between them, meaning that they could actually be fertile and be able to conceive with other people outside of the couple. But this is for human, and why do we think it could be interesting for cattle? Oops, sorry, come on. Okay. It's because matings in the dairy industry are planned based on the potential of the parents to produce the genetically best offspring possible based on the breeding goal of the farmer. However, when you do that, you usually assume that all the matings have the same chance of success, which may not be the case due to pair incompatibility. This is not always an all or nothing situation. We could have genes with a smaller influence on the compatibility, but this compatibility could impact the genotype representation in the next generation. But before being able to estimate the impact of gametic compatibility on our estimation of fertility, we need to understand the biological mechanism of it. No matter the species we are speaking of, we know very little about gametic incompatibility, so we wanted to investigate it and its genetic background in dairy cattle. To do so, we first needed to identify genomic regions associated with this incompatibility. To investigate this, we used transmission ratio distortion, TRD, which is a deviation from the Mendelian inheritance expectation. Maybe you remember from high school or undergrad in your biological class, the Punnett square, the combination square, where you have two parents, one here, which is each two gametes, the big A and the small a, with normally 50% of chance of transmission for each of them that you can see here, which will create 4 potential genotypes within the offspring with an equal chance, here 25%. This is Mendelian loss of inheritance. When you have TRD, one parental allele is preferentially transmitted to the offspring. So those 50% of chance here can be anywhere between 0 to 100%. And the difference from the 50% is interpreted as a TRD signal. With the case of gametic incompatibility, more than the transmission part, we were actually more interested in the allelic combination within the offspring. And that's why we adapted a TRD model to our needs. But before going to the complicated way, I will start by explaining the principle of a normal allelic TRD model. To summarize and simplify as much as possible, a normal TRD model relies on the probability of inheritance of a given allele from an heterozygote parent. Why heterozygotes? It's because homozygotes are not informative. They will always transmit the same type of allele. Here you can see the probability of transmitting the allele A1, which is going to be 50% plus the TRD effect that we are going to call here alpha. For A2, it's going to be 50% minus alpha. This is intended for one parent, so you have to do it for the other parent as well, meaning that the TRD effect can be sire and dam specific. Then the probability of the genotype of each offspring can be defined by the parent genotypes and by the TRD parameters. Here I'm just presented you one of the possible offspring genotype out of the 4 possible. Remember the Punnett square. Here it's going to be A1, A2. And to obtain this genotype within the offspring, you have 2 potential combinations from parents. The sire giving a A1 allele with the dam giving A2, or the sire giving A2 and the dam giving A1. This equation has to be defined for each of the genotype combination, but very largely, these are the TRD principle. For our study, we adapted the model. So once again, we'll only present one combination out of the 4 possible to just keep it simple. In this case, I chose the combination A1, A1. So A1 from the sire and A1 from the dam. You can see that the first part is the same as what we previously saw in the previous slide. So that's the TRD effect, the direct TRD effect. But there is an addition of a beta term. Beta, it's the interaction of allele within the offspring generation. There are 4 potential genotype combinations, so 4 terms. But only one combination happened within the offspring. In this case, the combination A1, A1 that you can see here. Therefore, the model includes the probability associated with the present combination times the probability of not having the other combination. This model was run using the TRD scan software created by Samir, Dr. Hilla Hussein, that did his PhD with us some time ago now, which pretty much consists on a scan of the genome using a SNP windows to identify genomic regions with TRD signal. For computational reasons, we couldn't scan the whole bovine genome with the adapted model, so we decided to just scan regions that were already previously identified with direct TRD effect. Our dataset consisted of more than 430,000 50K SNP genotype of Canadian Holstein cows, animals, composing 280,000 parent offspring trios. We decided then to filter the regions based on the magnitude of the effect to keep only regions with strong genotypic interaction effects. Out of this, we extracted the positional genes for functional analysis, which were performed for both gene ontology terms and cake pathways. For the functional analysis, I will explain you a bit of the interest of each of them. So for the network analysis, we were looking at how the genes and the proteins out of the coded by those genes were interacting together and which biological pathways were associated with those interactions. For the overrepresentation analysis, the general approach is to identify biological pathways associated with our genes that are more present in our dataset in comparison to what you would have gotten from a random sample of genes. For the guilt by association analysis, it is about identifying genes that we will call prioritized or guilty by association genes, which are sharing a functional profile with candidate genes for the phenotype of interest. As they are sharing a functional profile, they are very likely to be a candidate genes as well and to play a role in this phenotype. To identify the candidate genes within our gene list, we relied first on their association with keywords, such as fertilization and sperm-egg recognition. Then we confirmed them with the literature, and with that we were able to perform the guilt by association analysis to identify prioritized genes. Now to the results. We got 422 regions containing around 2,000 assembled genes ID. For the network analysis, this gene list created 13 networks. One continent, so meaning a network containing a lot of genes interacting together. In my case, I had around 400 genes. And 12 islands, so which were a network composed of 10 to 5 genes interacting together, so much smaller. We focused on the biggest network to understand the mechanisms behind gametic incompatibility. As you can see here, discussing the network itself is a bit complicated, so I'll just give you a summary of the pathways associated with it. We got 120 significant cake pathways and 144 GO terms divided in the biological pathways, molecular function, and cellular component terms. To summarize, most of the functions were relating to immunological and cell signaling functions. And there were actually very little pathway that we could directly link to reproduction. For the overrepresentation analysis, we got relatively similar results to the network analysis. There were sadly only one analysis that was significant for a false discovery rate of 5%. It was for the GO term linked to the cellular components. Before speaking about the results themselves, let me explain you what you see on the table. The column table total is the total number of genes within the whole genome that is associated with the terms that you can see. Expected is for the number of genes within the network that will be associated with the GO term if your gene list would have been a random sample of genes. For hits, it is the actual observed number of genes within the network for your term. Then you have the ratio of overrepresentation, so hits over expected, and the false discovery rates for 5%. For the result now, we had an actual overrepresentation of the entities linked to the genetic material, such as the chromosomes and the endoplasmic reticulum. Even if most of the other results were not significant, we still got quite some insight on the important mechanism behind gametic incompatibility by looking at them. So when we looked at the overrepresented Kirk terms, Generally, the enriched pathways were related directly to immunologic functions or the dysfunction linked to diseases, as you can see in this list here. Most of the pathways were linked to autoimmune diseases. When we looked a bit more in depth on that, we found that there were a lot of genes coding for the major histocompatibility complex, MHC, which is also called HLA in human, which is a set of highly polymorphic and polygenic genes coding for self and non-self recognition, which is actually quite interesting considering gametic incompatibility, which relies on cell interactions, and more precisely on the modulation of this process to accept the sperm cells, which are foreign to the female reproductive tract, and the interaction of the gametes, which are of different origins. So, and as well, because genes of the MHC were also known to be part of the mate selection at both the individual and the gametic level. When we look at the guilt by association analysis, we got to identify 12 genes associated with gametic interaction and fertilization. Those genes created really a strong functional profile associated with gametic incompatibility. I will not go too much into details, I don't want to bore you, but I would just like to mention the presence of these genes here, CD9, which is one of the very, very rare genes that is accepted to be associated with gametic compatibility in the literature. The other genes were either on the suspect list or known to be related to a very close mechanism. The next step of the analysis, we got 4 genes with a statistically similar functional profile to the candidate genes. The 2 first genes are known to be part of the immune system. You may recognize, for example, the name of the interferon gamma here. MET is linked to an abnormal gametic genesis and embryo implantation, and ATP2B4 is playing a role in the gain of fertilization ability of the sperm cells, which is bringing me to something interesting to notice with this analysis. Most of the genes could be grouped into 3 categories based on which fertilization mechanisms they influence. The first one will be the genes linked to the gain of fertilization ability of the sperm. The sperm cell have to go, and there are different reactions within the female reproductive tract to gain the ability to fertilize the egg. You have, for example, the acrosome reaction or the sperm maturation, and those events have to happen at a specific timing for the sperm cells to gain their fertilizing ability. The next step will be the gametic interaction, so sperm cells and oocyte recognition and fusion. And here we have the example of CD9, which plays a role in the binding of the gametes. For the last category, it will be for the genes relating to the female immunoacceptance, so the acceptance of both the sperm cells but also of the embryo. And most of the genes that we found and put within this category seem to play a role on both sides. So they were pretty much genes important to the immunomodulation linked to reproduction. So with those analyses, we got to understand better what is gametic incompatibility and which biological pathway it is relying on. We identify some genomic regions and more specifically a few genes that are coding for this mechanism. With the information and with further investigation, the identification of compatible haplotypes per would be possible. So we could potentially predict the chance of success of a specific mating, which could help improving mate allocation in dairy cattle. And if we want to look even further, we could maybe select for more compatible individuals. The take-home messages are that not all matings have the same success rate for other reasons than infertility of one individual. We showed today that using a TRD model with a genotypic interaction term allowed to identify regions and genes associated with gametic incompatibility. We also found that the involved mechanisms are linked to immunology and cellular signaling processes, meaning that gametic incompatibility is also a thing in dairy cattle, and by considering this compatibility, we may be able to improve mate allocation in the dairy industry. But this is not for today. We will need more studies to investigate the impact of gametic incompatibility on our current estimation of fertility. This was one approach to improve fertility as we know today. Another approach to improve it is by looking at other potential traits that could be closer to the biology of the animal than what we have right now. I will speak about one of these possibilities, the size and position score, called SPS, as a potential novel trait for genetic selection. Why do we need a novel fertility trait to help us breed for better fertility? More than 30% of the cullings in Canada are due to reproductive issues. The number has only been growing in the past decades. On top of the obvious welfare issue, it is also an economical loss, as most are involuntary cullings. They are not planned within the herd management. More, the producer also lost the genetic of those animals, which affects the current herd generation, but also the next generation, as some high genetic merits animal for production, for example, are not part of the reproducer's pool anymore. They are not parents anymore. And to solve this problem, in Canada, we started selecting for fertility in 2005. But improvement is really slow. We stopped the decline, but we did not have an improvement of the traits yet. And there are two main reasons cited for this. The first will be the low heritability and reliability of the traits. You can see it in the table here, for example, which are the common reproduction traits we have in Canada, which range between 1 to 6% for heritability, which make them relatively difficult to be selected for accurately and efficiently. It's not impossible, but it will take a long time to get a significant response to selection. The second reason, which is coming from the unfavorable genetic correlation with production yield traits. We are heavily selecting for them, and this is creating a bit of an economic impasse here. That's why today, with the technology and the knowledge that we accumulated since 2005, we can, as researchers, investigate new ways of measuring and selecting for reproduction. Potential new traits with better parameters, so better heritabilities or an absence of antagonism with production, for example. In our case, I should say, we propose a novel fertility trait called SPS, which is a physiological indicator of fertility, for which we estimated the first genetic parameters to determine its potential as a trait for genetic selection. But let's start by defining what is SPS and why it is interesting. The size and position score, SPS, is a simple morphological measurement of the reproductive tract. You can see it on the side. It consists of 3 categories based on the size of the tract. So for a cow with a small reproductive tract, she will have the cervix and the uterine horns resting on the pelvis. For a cow that will be scored 2, you will see that the cervix is still on the pelvis, but the uterine horns fell within the abdominal cavity. For a cow scoring 3, you will have a reproductive tract which is much larger, much longer, with cervix and horns within the abdominal cavity. Those scores are attributed by transrectal palpation when the animal is not pregnant. So in the study that I'm just going to present you in the incoming slide, they measured SPS at insemination, for example. So I think you will be agreed with me. This is a simple trait, but what is the interest of it? To answer this question, I will present you the result of an American study from 2017 that was made on around 2,000 cows. And the results from this study were actually consistent with a more recent physiological study of SPS that was carried out at the University of British Columbia. First of all, with graphic A, we can see the breakdown of each of the score by lactation. In the first lactation, after a single calving, there is a predominance of score 1, so a relatively small tract, and a low representation of score 3. After a second calving, we can see a decrease in score 1 for score 2 and a very slight increase in score 3. In lactation 3 and more, we lost the dominance of score 1 for the score 2, and we have a larger proportion of score 3. So not really surprisingly, with more and more calving, the reproductive tract gets larger and larger. But why is it interesting? To answer this, I invite you to look at graphic B. You can see the chances of pregnancy after an insemination for each of the scores. A cow scoring 1 will have 43% of chance of being pregnant after one insemination versus 36% for an SPS 2 cow versus 27% of chance for a cow scoring 3. This implies that a young cow in first lactation scoring 3 will be statistically less fertile than an older cow scoring 1 but with many calvings. So this graph explains the physiological interest of SPS. We want more SPS 1 cows. And to reach that, we may want to use genetic selection. So I will now present you the genetics aspect of this trait with my work. I had the chance to get a dataset that was collected for a physiological study of SPS by Dr. Ronaldo Seri's team at the University of British Columbia, where from 2017 to 2020, the cow from the research herd were scored for SPS using transrectal palpation, and this from calving until conception in different parities. So one cow could have a unique record or multiple records within and or across lactation with potentially different SPS results. It was composed of more than 3,000 records from 490 cows. And to summarize a bit the complex structure of the dataset, I had 213 cows with multiple records within one lactation, 248 cows with multiple records within and across lactation, 5 cows had one record but in different lactation, and finally 29 had a unique record. There, I fitted an animal model to this dataset. Because we had a small but complex sample, we kept the model relatively simple. The phenotypes were explained by the fixed effect of year season, year being 2017 to 2020, and season being defined every 6 months. 4 stages of lactation. So the first 21 days in milk as uterine involution, and then I used the usual 120 days in milk for early, mid, and late lactation. And I finally corrected for the parity number as a linear and quadratic regressions. For the more common part of the equation, I corrected for the random animal effect, as well as the within and across lactation permanent environmental effect, as we had repeated record. Due to the novelty of the traits, we compared threshold and linear models for SPS. Theoretically, the threshold model is advantageous because it respects the categorical nature of the trait. However, the linear model is usually preferred as it is both less complex, less computationally demanding, and usually result in the same animal ranking, so same results. We compared the two models using the DIC, the Deviant Information Criteria, which use the goodness of it and the number of parameters fitted. And we also use the EBV rank correlation to determine which model we will go with for the estimation. And the estimation were done using a Bayesian approach to perform the variance component estimation, meaning that we got all of the variances and repeatabilities. Additionally, we looked at the correlation between SPS and the Canadian Indexes of Selection, LPI and ProDollar, as well as with the genetic correlation between SPS and trait that we are currently selecting for in Canada. Those traits were chosen relatively simply for the economic importance of the production traits. We were actually also interested to see if we had the usual trade-off between production and fertility between SPS and production. Then we looked at reproduction traits for the biological relevance. They are the commonly used indicators of fertility in Canada. All of this was fitted in a bivariate analysis using the SPS model I presented earlier and the models from Dr. Oliveira's paper, which are the model commonly used in Canada, but I just adapted it to the dataset. So for example, we didn't have herd effect, so I removed it from the model. Now to the results. For the comparison of the linear and threshold model, you can see that the threshold model had a lower DIC, meaning that it has a better fit. It's not really surprising as SPS is a categorical trait. However, when we looked at the correlation between EBV ranking from the two models, we saw no difference, 99%, so we have the same ranking. This implies that we can use a linear model and get similar results to the Distortion model. Because linear is usually preferred in animal breeding for diverse reasons, such as the easier interpretation of the estimates, we went further with it. Now we can move on to the real results. And before even discussing them, I would like to remind you that the estimate that I'm going to present you were made with a dataset collected for a physiological and not a genetic study. We had 500 cows from a single herd, and the scores were collected over the entire lactation. Normally, for genetic analysis, we would want more cows, more herds, and a standardized phenotype collected at a specific time, which is not what we had here. So the estimates are true for my sample, but can be impacted by the situation and will have to be confirmed with a larger scale genetic study to check for the population. But let's return to the results. For the habitability, we are at 11%, which is relatively low, but I remind you that the maximum we have today for fertility is 6% with the open days. So here we have an improvement with SPS. For the repeatabilities within and between lactation, we are at 24 and 13% respectively, which is once again low, but here I think it's also very impacted by the dataset that I used, so I will not discuss those numbers much further. For the correlation with the selection indexes, we had minus 0.23 with both LPI and predator, which is once again low, but here it's favorable. As SPS1 is more desirable than SPS3, the optimum is at the minimum, while for the selection indexes, it's the opposite. We really want the highest value, that's the best. This opposite direction means that the negative correlation is favorable. The best ranked animal for the indexes of selection tend to have an advantageous SPS score as well. For the genetic correlation with the current traits, we will start with the production, so milk, protein, and fat. And you can see that the correlation are all neutral, so they are not different from 0, which implies that the selection of SPS would have no impact on the production traits, and we can select for higher yields without deteriorating SPS. This is a big advantage since we have lost the usual antagonism between production and reproduction here. For the reproduction traits, the first 4 traits, age at first service, calving ease, calving to first service, and days open, it is as for the production traits, we have correlations that are either very close to 0 or not very different from 0, or not different from 0, sorry. There will be no impact on selecting for SPS on those traits and vice versa. For the others, first service to conception, non-return rate, and number of services, we have relatively high correlations. For non-return rate, it is similar to what happened with the indexes of selection. We have the optimum in different directions, so negative correlation here is favorable. Selecting for one would improve the second trait as well. Contrary to what we had with IPV and predator, first service to conception and number of services have a positive correlation, which is favorable. Those traits and SPS have the same optimum to the minimum, so selecting for SPS would indirectly select for these traits and vice versa. If we want to go a little further using the theory and the number I just presented you, the selection of SPS would give an indirect answer to the selection for these 3 last traits, which would be more important than if we were directly selecting them. But of course, this is theory and has to be confirmed by a practice with a study. To conclude a bit on this genetic correlation, selecting for SPS has the potential to be more effective than what we currently have, and selection would not be slowed down by an antagonist with male production. Finally, for this project, the information I would like you to remember is that we have results for SPS which are preliminary, so to be confirmed with more cows, more herds, genotypes, and all the things next to that, but which are very promising. SPS is an easily measurable trait with a better habitability than what we currently have, as well as favorable correlation with most current fertility traits. Therefore, we have a potential to select more efficiently for fertility. We also have the absence of antagonists between SPS and production, which implies that the selection of one will not prevent or slow down the selection of the other. We have here a novel trait with the potential for a more efficient selection of fertility. So to conclude more generally, I presented you two different approaches to get better fertility in dairy cattle. One was a novel concept in livestock, the gametic incompatibility, which could help the mate allocation and could improve our current estimation of the fertility ability. And the second part was on a novel trait, SPS, which presented promising parameters that could potentially improve the selection of fertility. So I hope that I successfully presented you and interested you in some novelties for the genetic improvement of reproduction in dairy cattle. And with that, I would just like to thank all of the funding agencies and supporting partners that made those projects and mitosis in general possible. And I would like to thank you for your attention. And please feel free to ask any question. I will be very glad to answer them. And you can also reach me by email if you want.
Thank you, Audrey. So if anyone has any questions, please feel free to raise your hand, and I'm just about to change the meeting options so that you'll be able to turn on your cameras and mics. Perfect. So questions for Audrey. I have one quick question, Audrey, and I'm pretty sure I asked it before. If in a beautiful scenario this gets implemented, do you think it's going to be the responsibility of vets to collect this data, or do you think it'll be simple enough for producers with it, I'm assuming, some sort of training to be able to determine SPS 1 versus SPS 2 versus SPS 3?
So actually, SPS is easy to measure, like it's a transrectal palpation, so it's not a very big deal and it's not heavy in the management and stuff. However, it really needs to be consistent over all of the cows and over different herds. So that's why if producers are measuring it, we may have some bias there. So I was thinking, like when we were discussing it on how to develop the treat and everything, we were more actually into asking potentially the insemination technician to record it, because they're used to do transrectal palpation during the insemination. So it will actually not be a big deal for them to just spend 5 seconds more on each cow for the scoring. And they really have they could be easily trained to keep the standard between each of the measurements. So I think that would be the best solution. Asking vets to do it will also be a solution, but that will be also maybe something to consider. I think insemination technician would potentially be the best option.
Okay, perfect. Any other questions for Audrey? Yes, we have one in the chat from Guilherme. Audrey, congratulations for the presentation. I was wondering why the position of the reproductive tract would benefit fertility outcomes. Thanks.
That's an actual good question, because SPS is relatively new. We actually don't, well, we have a hypothesis, of course, but knowing why the position plays a role, still a bit, maybe because it is, there is no clear answer there. If I had to answer that in my way of thinking, that will maybe due to the markers that is present within the tract. So with more calving, you potentially have more scar tissue within the female tract. And to detect the conceptus, you will need more markers to be excited, I'll say. So to give the signal that, yes, there is an embryo, so don't start a new cycle and stuff. That could be one hypothesis, but there is something up to discussion, I think. And if you have an opinion there, I will be very happy to hear it, because that's really something still up to discussion.
Leading on from that as well, Audrey, I might have one more, just thinking off the top of my head. And could you almost treat SPS as an internal conformation trait, like within the body conformation trait in terms of like shape and size?
Well, it is a conformation trait.
Okay, so
it is a morphological trait, so it is a conformation after all. I didn't present it, but we did look also at the genetic correlation with some rump conformation traits.
Yeah, that's what I was going to ask, yeah.
We did look at this. The problem is that most of the rump traits have intermediate optimum, so you don't want extremes, which makes a linear correlation very difficult to interpret. We do see that the correlation are quite high, so this seems to have an impact from one to the other. So there's this. We know as well that the correlation with the body condition score is quite high, so that could be also a thing. For me, I don't know if everyone's going to be agreed here, but for me, it is a morphological trait. It's a morphological trait of SPS, and that's why it's actually so interesting, because we lost the idea of management. Yeah. So the trait could be affected by diseases and stuff. So you could change score if cars and arthritis or stuff like that, but it will not change if she's producing like 9,000 kilos of milk or 12,000. That will not affect the size of the uterus. That's what you expect from a morphological trait. And I think, for me, that's why it's so interesting to look at this for a fertility trait.
Yeah, that's from my history of doing fertility traits, yeah, to see the fact that management has been taken out completely, even in terms of like producer decisions and when to breed animals is completely taken away, while so many fertility traits that we're looking at now is producer dependent. So yeah, very exciting stuff.
Yeah.
Any other questions for Audrey before we finish up? Yes, Eduardo.
Try to get my mic working here. If you hear a baby, it's just he's going crazy now after lunch, I guess. Great presentation, Audrey. Really enjoy it. I was going to ask you about your first study. That's really interesting. You talk about gamete compatibility. I think that was the term that you mentioned. So I lost the beginning of your presentation. It took me a while to connect, and I was trying to understand what that actually means. So you show a group of genes that I'm assuming some of those is SNPs, depending of what is in the cow or in the bull, they might not be compatible for embryo development or for fertilization of the egg. Is that what it is? Can you explain that to me? Sorry if it's a naive question. I just I'm trying to understand what it means.
No, there's no worry there. Gametic incompatibility, as I define it, will be an incompatibility of the gametes to interact together. So it could be actually, in the way that we studied it, either the sperm cells that cannot interact with the female reproductive tract to get proper fertilization, to reach the egg, etc. Or it could be also a problem of interaction between the gametes, so a problem of fusion or a problem of development of the embryo in early stage and things like that. That will be a gametic incompatibility, and the genes that you've seen here are shown to play a role in those mechanisms. And it seems that I presented one example, the CD9 genes. Here we know that if this gene is mutated, there is a mutated allele for this gene that is just not compatible. So you will not have any fertilization with this allele. but the other genes could be just, it is compatible, but at 10% instead of 60%, let's say. So that's
so less efficient, let's say, in terms of generating a live pregnancy will be less efficient. Okay.
Yes.
Yeah. It's very interesting. Yeah. Maybe we can meet and talk more about those genes sometime.
I would be very happy to do so.
Yeah, thank you so much, Audrey.
Thank you.
Eduardo,
Thank you,
just to jump in. I think I think that would be a great idea. No, so it would be really nice to have a follow-up of these genes in another study, and I think you are in a great position maybe to to take this knowledge to the next level. I don't know if Eduardo was able to hear me, but anyway, it's a great idea. And congratulations to Audrey.
I wasn't sure if you were talking with me or with Audrey. I'm sorry.
No, Eduardo, I just said that would be a great idea that you could meet with Audrey, because I think there is some candidate genes there that you in your work could take to the next level that we need really to prove the certain aspects of the gametic incompatibility. So it would be great, because Audrey is finishing up, right? So it would be good that somebody else would have interest to follow up on this discovery.
Yeah, absolutely. We'll be in touch for sure.
Okay, and again, Audrey, congratulations, great presentation, very clear, very didactical. Well done.
Thank you.
Great, thank you, Flavio and Eduardo. Any final questions then? I'm not seeing any. So once again, Audrey, I'd like to thank you, just like Filippo and Eduardo, on a fantastic presentation. And thank you everyone for joining us. We'll see you all next week.
Thank you.